All right, so today I wanted to talk about this preliminary paper, this very new paper. It's so new that it has this accelerated article preview here that talks about the genetic mechanisms of critical illness in COVID-19. And we can see here that the accelerated article preview was published just 2 days ago as I'm recording this on the 13th. And I wanted to go through some of what this paper was talking about. And there's actually a different form of it here, which I believe is an even earlier version. I have both of these linked to in the description below if you wanted to go through them yourselves. But I am going to go through some of these data here. So one of the things that they found was and that they're most interested in are these four genes here. And so the OAS3, this tyrosine kinase 2, this DPP9, and this interferon receptor 2. This gene here is not, they don't seem to be as interested in. This has more to do with things like development. That should be an OP development and things like that, and cell growth. But these four are all known to be involved in immunity and immune responses. And so this is the table that they were looking at. I'll go through the data in a moment, but I first wanted to go through what each of these different genes actually code for. And so the first one here I'm going to talk about is the OAS genes. And so this, as I said, is involved in immune response, particularly to viruses. And so interferon, which is this INF here, comes and binds to a receptor, and this will activate the transcription of the OAS gene, which is then involved in attacking double-stranded DNA. So the RNA encodes proteins activated by double-stranded RNA, which is the viral RNA. So this comes together. This turns ATP into this 2,5 version of the ATP, which then goes and activates these RNase enzymes. So RNase is just an enzyme that breaks down RNA. And so this RNase L goes and starts breaking down the viral RNA. It will also break down our own RNA. So when humans when a gene is expressed, it generates mRNA, and this can also break down our own mRNA as well as the viral mRNA. But this is sort of the target that it's interested in, and so that's how this OAS that we saw up here, this OAS3, which is one type of OAS, is involved in the breakdown of viral RNA. So the next one I'm going to look at is this DPP9. So this one I had a much harder time sort of pinning down exactly what it's for. It seems to have a bunch of different functions in the body, but one that I found is that it's involved in the processing of epitopes that our immune system can then present on the surface of the cell to sort of signal to other cells to look for that particular protein and attack it. So the proteasome breaks down the protein into these small pieces, these small peptides, and then these can be further processed by this DPP9 here, into here, which just breaks off small portions of the protein near a proline residue. And so over here it shows the DPP9 breaking down this SYK protein, which is a type of kinase protein involved in immune response. And um, I'm not going to really talk about that one, but it's similar to another one that we have up here, which is this tyrosine kinase 2. And I'll talk about that at the same time as this interferon receptor, which is down here because the interferon receptor 2, which we have here, and the tyrosine kinase 2, which we have here, are sort of involved with each other. And tyrosine kinase 2 is a type of these JAKs, which are Janus kinases. So a kinase is an enzyme that phosphorylates other proteins. So it takes the phosphate from an ATP, and then this will turn into ADP by losing one of its phosphates, and we'll have a protein here that is being phosphorylated. So then it'll be a protein with a phosphate group attached to it. And so this is how you can turn different proteins on and off. And so these are protein kinases, and they're called tyrosine kinases because they specifically phosphorylate the tyrosine residue. And anyway, I'm not going to go too much into that biochemistry, but just know that the interferon receptor, which these interferon proteins bind to, and that causes a conformational change in this receptor, which then activates these tyrosine kinases, which will then phosphorylate other proteins. And then we can see over here we have these signal cascades. Once again, we have our interferons coming in here, and the interferon receptor 2 and our tyrosine kinase 2, interferon receptor 2, and these Janus kinases, which are very similar to the tyrosine kinases. We see that we have these signal transductions going through. I'm not going to go through all of them, but just notice that they have these stat proteins here, and this is why it's called often called the I can write it better than that, the Jack stat pathway, because we're using these JAKs and these stats, these various stat proteins. And anyway, I won't go through all the signal cascade of this. I do have links in the description below to papers that all these figures are taken from, but sort of the big picture here is that they go into the nucleus and they bind to these things called interferon response elements and things like that, and activate these different genes, which then code for proteins that go on to do other things. Particularly, what we're interested in here are these antiviral responses, and so we can look at that down here. And so when interferon binds to the cell, it actually activates all of these different proteins that they just have as stars here. And what they show on this figure here is these different proteins at different places in the viral life cycle. And these are the parts of the viral life cycle that these are actually inhibiting. And so we see here our OAS, which I said before was breaking down the viral mRNA. So this is the viral mRNA here. And so these proteins here are all breaking down that mRNA before it can go to be translated. But then we might have this protein kinase R here inhibiting the translation. And so we can see the interferon turns on the expression of all these different genes. And so we have genes here that can can inhibit the what's called the egress of the viral particle, and we have this protein here, which sort of inhibits the assembly of the viral particle. And anyway, I'm not going to go through all of these. I just wanted to show that when we do this this interferon cell signaling here, that what it's turning on with these genes are these various proteins here that inhibit the viral life cycle. And so that's sort of why interferon is important, is for turning on these proteins that inhibit the viral life cycle, and for signaling to other cells to do the same. Right, and so what this article was doing was trying to find what genes are or what variants of these genes, so things that are called SNP, or single nucleotide polymorphism. They wanted to find what SNPs seem to be present in people who got severe illnesses with COVID-19. And so they use what is called a genome-wide association study, or GWAS for short. And so what that does is they sequence the genome of patients who had the disease, and the genome of people who did not have the disease, and then sort of compare them and see what sorts of SNPs are overly represented in the population of people who had the disease, that are not overly represented in the people who did not have the disease. And so they compare the people who had the disease with the people who did not, and find what SNPs seem to show up more often in people who had the disease as opposed to the people who did not, or just non-patients, sort of the general population. And so what they do then is they use this thing that's called a Manhattan plot. And this has the different chromosomes here across the bottom. So humans have 2 copies of 23 chromosomes. So we have 46 chromosomes total, but there are 23 copies, which, if you've had your genome done by 23 and me, that's why they have the 23 here, because of the 23 types of chromosomes we have. And anyway, they're looking at what's called the negative logarithm of the p-value. So the p-value is basically just a statistical tool used to show whether whether when you see some difference between things, how much you can attribute that to chance and how much you can attribute that to some causal factor. And so they were looking at the OAS, the DPP9, or the tyrosine kinase 2, and the interferon receptor 2. And so if they have dots above this red line here, they're saying that that the p-value is such that that shows that there is some causal mechanism, some causal factor involved. That there seems to be that this gene here, so this DPP9 at this spot here, is above that red line. So they're saying there must be something interesting about this gene, about this the variant of this gene, rather, the particular allele or SNP of this gene being overly represented in the diseased population, and that must mean that there is some reason that this gene has some causal influence, or that the overrepresentation of this SNP is statistically significant. I think would probably be the most accurate scientific way of talking about it. And so they were interested in these four genes because they weren't interested before they did the genome-wide association study, but when they found this in their genome-wide association study, they sort of focused in on these four genes because of, as we talked about earlier, they are involved in the interferon response and the viral immunity. You can see some of these other genes are also overrepresented, including this FYCO1 gene, which no, I didn't put that on here, but I think I have that open on here. So the FYCO1 is one of these type of genes called, they call them RUFY genes because they have this RUN domain and this FYVE domain. And these are, so we can focus in down here, this has the FYCO1 here. And these are involved in carrying these endosomes around the cell. And this shows the RUN domain is binding to these cytoskeletal structures, and then this FYVE domain is binding to the actual endosome, and then this can sort of walk along the cytoskeletal structure and sort of transport this around. And so this is involved in moving these phageal lysosomes around and these other endosomes, and so this is involved in phagocytosis, which is another immune response. And so the paper didn't really focus in as much on this one, but this does seem to be involved in phagocytosis, which is an immune response. But they did find that these four genes are also overly represented in the diseased population. So now we can go back to this table here, which these are the four genes they were interested in. And so these are the codes for the SNPs here. And so they found that at these particular sites, these SNPs, you can have this allele, which has an adenine at the site on each of these, rather than a guanine or a thymine, which are the non-risk alleles or SNPs, and these are the risk alleles. These are those p-values that they use to plot on this Manhattan plot here. And so I actually went into my 23andMe raw data and looked for these SNPs in my genome, and I found that I have the A or risk gene in my DPP9 and OAS3 genes. So I'm heterozygous for the risk gene. So I have the risk SNP in one of my chromosomes and the non-risk in my other chromosome for the DPP9, which was this one here. So I have the A in one version of the chromosome, one copy of the chromosome, and G in the other, and same for the OAS3. I have the A and G. So heterozygous. So as I said before, we have 23 chromosomes, but for each one we have two copies, and so uh, on the DPP9 is on the 19th chromosome, and the OAS1 is on the 12th chromosome. And so for the OAS1, my 12th chromosome, so I have two versions of that chromosome. On one of them, I have A, and on the other one, I have the GSNP. And then so for the DPP9, which, as I said, was on the 19th chromosome, it's the same thing where I have the 19th chromosomes are smaller than the 12th. The chromosome numbers are ordered such that it goes from largest to smallest chromosomes. Then they kind of put the X and Y chromosomes at the end, the sex chromosomes. But anyway, my 19th chromosome, one of them has the A risk SNP and the other has the G non-risk SNP. And so I looked further into these two SNPs, and both of these are actually intron, so they are found in the intron of the gene. And so what an intron is in a gene. So you have your DNA, and you have a section of the DNA that's the gene. But only small parts of it, called exons, actually code for the protein that this gene expresses, and so the parts in between are called introns. And when the gene is transcribed, it transcribes the whole thing, including the introns. So the pre-mRNA will have all of it. But then the introns will get spliced out, and you can have what's called alternative splicing. So you might have exon 1, 2, and 3 go into the protein coding mRNA, or you could have exon 1, 2, and 4 go into a different or alternative splice of that mRNA, which will code for a different protein. Oftentimes similar proteins. A lot of times this alternative splicing is dependent on the type of cell. So you might have this version of the protein in, say, like the liver cells, but then you'll have this, and it's sort of the same protein, but it functions a little bit differently, and maybe this will be in muscle cells or something like that. And so that's oftentimes what this alternative splicing is doing. And so it seems a little counterintuitive that this SNP, the single nucleotide polymorphism in the intron, would change the protein. But what happens is the alternative splicing requires all these various factors. I'm not going to go through the whole splicing mechanism, but we see here that that some of these proteins have to bind onto the intron, and so they're going to look for certain recognition motifs in the intron, certain small sequences of the A, T, G, and C. So the different nucleotides, different sequences of these nucleotides they'll bind to. And so if you aren't binding these as tightly, or if you're binding them too tightly, or just binding them in different places and stuff like that, you might change from one type of alternative splicing to another, or it might slow down the splicing or speed it up, or have various effects on the splicing. And that's how a mutation or an SNP in the intron can lead to different functionality in the actual protein. And that's why having the different functionality in these proteins involved in the interferon response and in the immune response can lead to differential outcomes in the actual disease. And so having these risk SNPs in these genes can lead to differences in the alternative splicing. At least in the DPP9 and OAS3, I didn't actually look up the TYK2 uh, or the interferon receptor 2, uh, mostly because I didn't find these SNPs in my genome, and so I guess out of laziness or maybe selfishness, I just didn't look those two of them up. But anyway, this can, as I said, lead to different disease outcomes because of the differences in alternative splicing, or alternatively, if it's an SNP in the exon, then that's a little more straightforward. If you're coding for one particular amino acid, and you have an SNP that makes it code for a different amino acid, that's going to change the functionality of the actual protein. And so that could also be a factor in why having different variants of these genes, different alleles of these genes, end up having different outcomes in the disease severity. And so anyway, I just wanted to go through this new paper that was talking about the different mechanisms of critical illness in COVID-19. Um, yeah, I think that's all I'm going to talk about here, so I will see you in another video.